Good morning, everyone. My name is Nattanit Kulviwat. I am fourth-year medical student from KMITL, and my advisor is Professor Dr. Shear. So today I'm going to present you about aspect of shear stress on morphological and functional property of H-back5i cell in the newly developed blood-brain barrier on-chip model. Neurological condition is the #1 leading cause of disability of disease burden worldwide. Around 3.4 billion individuals had affected by the condition, and this leads to 443 disability at just life year. It has effect on the individual level, economic level, and healthcare sector level, and it has cost more than 1.7 trillion per year. So all in all, the pathological of the disease are different. However, one of the common factors is disruption of the blood-brain barrier. So the blood-brain barrier is a selective barrier of the neurological lumen, and it has tight junction, so it controls the movement throughout the cell. So by having more understanding about this, we could have more understanding about the disease and drug delivery. However, there are several limitations of the current experimental model. For example, in vivo model that rely based on the cell culture process in 2D model. So it has lack of physiological complexity, so it is inadequate to represent the disease microenvironment compared to in vivo model, which has more complexity. However, due to the variation between species to species, so it has limited human relevance. So the organ on chip is developed based on those limitations. So what is organ on chip? Organ on chip is the technology that combines the cell culture as well as the microfluidic technology to mimic the vasculature, blood vessel, and lymphatic flow of the cell. So it enables us to study more on the organ function, mechanism of the disease, discovery the new drug. So there are several factors that affect the microenvironment. For example, the chip design, surface modification, flow rate, and flow time. So in this study, we want to investigate the effect of shear stress on the newly developed blood-brain barrier. In terms of the functionality, we measure the permeability, and the physiology, we measure the cell morphology and expression of tight junction protein, so not occludin 1. So here is the chip architecture. So the chip is made of polydimethylsiloxanes or PDMS, and the oxygen plasma treatment will be treated the chip, and the chip will be coated with the collagen. So there are inlet and outlet of the chip, and there are 1 perfusion channel and 2 leakage channel. Between perfusion and leakage channel, there are micro channel that have width of 50 micron. So here is the picture of our chip, and here is the picture of the chip and the microtube for loading cell, for collecting fixed strand, and this is the chip under microscope, without cell and with cell. This is the overview of the experimental review. We will start off with the cell culture process. So we culture H-back5i cell, which is the human cerebral microvascular endothelial cell. This cell is usually used for studying endothelial function, cell trafficking, surface molecule interaction, and it has low permeability to 70 kilodalton fixed strand. Next, we will load the cell into the perfusion channel in both inverted and non-inverted side. Then we will study the chip in both stationary and dynamic condition. So in dynamic condition, we will generate the shear stress by inducing flow rate of 0.4 microliter per minute using silent pump, and the static condition will have no flow. After we flow the cell for 1, 3, 5, 12, and 16 hours, the image will be captured. And then after that, we will proceed for immunofluorescence staining for Z01 and DAPI for nuclear marker. So for permeability, we will introduce the 2 different molecular weight of fixed strand, which is 10 kilodalton and 70 kilodalton. After that, the image will be collected and analyzed by ImageJ, and the outlet media for fixed strand concentration will be used for calculation the fluorescence intensity, and apparent permeability will be calculated. Here is the result. As you can see, at 1 and 3 hours, the cell has the spindle-like morphology, and it starts to change into cobblestone-like morphology at 5 hours and persists until 16 hours. And compared to static condition, there are no difference in the cell morphology. And this is the result for SAT01 expression. You can see that there are higher intensity of the SAT01 in dynamic condition, especially at 16 hours, compared to the static condition. So next is the permeability. So when compare 70 kilodalton and 10 kilodalton, there are higher leakage of 10 kilodalton fixed disk strand in both static and dynamic condition. And it's confirmed by the apparent permeability that has been calculated. And it results that there are significant difference between 10 kilodalton and 7 kilodalton in dynamic condition, but not in static condition. And this is confirmed by the expression of the SAT01 and DAP. You can see in dynamic condition, there are higher expression of nuclear marker or DAP. So in conclusion, we show that by applying shear stress has significant influence on H-back5 cell morphology and functionality, which is evident by expression of SAT01 and endothelial permeability. So it's considered to be a suitable model for studying blood-brain barrier function and for study the central nervous system duct delivery. So here is our acknowledgement, and thank you for your attention.
Okay, thank you for a very interesting presentation. I'm wondering whether you get a conclusion of what would be the optimal flow rate that will not injure the cells, since you are trying to develop a model for blood-brain barrier, right? And yeah. I'm not quite sure is shear stress is what you don't want to have when you do experiment in the future. Is that why you are trying to study this?
Oh, it's because actually when you have like 2D model, you cannot introduce any shear stress into the model. So the blood-brain barrier enables us to induce shear stress into the model. So it is more complex, and we can study the cell culture under the dynamic condition. And the reason I choose the 0.4 microliter per minute for the flow rate is because we have varied the flow rate before into 1, 0.2, and the 0.4 is optimal condition that the cell is attached to on the chip.
Sorry, in the future, are you planning to use this as a model for, I'm not sure, it would be hypertension or something like that that would has the effect on the blood-brain barrier?
Yes, so we can adapt by changing the cell condition. For example, if you want to study on the hypertension on the brain, we can culture cell by inducing the hypertension into the cell and compare with the healthy one, and you can see the effect of the shear stress on the cell. And also, we can also add another cell, for example, co-culture with parasite, or changing into iPSC culture model. So it's more representable of the blood-brain barrier, but this one is just the foundation.